Well, thank you for inviting us here to this workshop. So I'll be presenting some of my research focus. Okay, so I'll just discuss 4 areas that I'm working on currently right now. First is the West Pacific Warm Pool, and everybody's very familiar with it. It's very important. Location. We're pretty lucky we have access to this area. Temperature is very important. Temperature reconstruction as well is very important, and there are many, many ways to do that. One that I'm working on is on molecular organic proxies using alkinone or TEX 86. This is very important based on organic matter. If the carbonates are not well preserved, or if you have siliciclastic material, and if you want to compare open ocean versus marginal seas. So the temperature reconstruction goes back to the molecular level because that's the only way that these organisms, one-celled organisms, could adapt to the outer environment. So that's the basis for the proxy. And for alkinones, they are recognized in 4 genera of haptophyte algae: Emiliana, Gepharcapsa, and Chrysotila, and Isochrysis. Maybe one problem with this in our area is that temperatures go beyond 28 degrees, and sometimes this does not work. So there's another proxy we could use. I've used this proxy and tested its veracity in Antarctic lake sediments, and it was very good for very cold temperatures, but it might be challenging for warmer temperatures such as what we have. So I would go for and test another proxy, the TEX 86 proxy, the GDGT, and we've done analysis for this in some parts of the Philippines. So it's based on archaea. This is their lipid membrane, and the one way that they could adapt to cold temperatures is having this cyclohexane in their structure. It's a very similar structure. If that one was based on archaea, this one is based on bacteria. If the previous one was used to reconstruct sea surface temperature, this one is used to reconstruct mean annual air temperature. So we have terrestrial and we have marine temperature reconstructions. Of course, like any other proxies, there could be pros and cons. It depends on your type of sample and where you are. So we've collected samples a little bit closer to the shore. We call us Pujada Bay. That would be the marine site. And then there's a terrestrial site, which is Lake Pinamaloy. So there would be comparison between terrestrial and marine area. Let me see. So this is where the Mindanao Current goes through. And then this is Pujada Bay. Some of that Pacific water impinged into Pujada Bay and gets recognized or recorded into the sediments. So these are our core samples. One thing good in this area is that the climate is almost very similar the whole year. Temperature is very similar, rainfall also very similar. So this is your temperature, average about 27.6 the whole year through. And annual rainfall, 150 millimeters, almost the whole year through as well. So this is our core. It's a short core. It's 1.4 meters long in the lake. This goes from, we've collected it around 2011. 1.4 meters at the bottom, it's about 1260 AD. So we've got about 700 years of data. Most of the problems in the Philippines is that we don't have a good instrumental record that goes back far in time. In this case, in Pujada Bay, that would be in Davao. And it only goes back around 1960s. So in terms of reconstructing temperature or paleo environments, this would be a problem. So at least for this core and for this site, we were able to reconstruct the temperatures based on TEX 86. We've gone down as far as 1260 which impinges into the warm, medieval warm period, which shows up here as a slight increase in temperature. After that, there's a little bit of decrease, and it's a little bit consistent. Thank you. This would be your little ice age, and I'm very happy to see this. In the deep tropics, we've got record of the little ice age. And we have to make sure of this because it's only a few degree, 1 degree and even less of temperature change. So that's why we have this error envelope to make sure that there's no analytical artifact. We've processed the samples in Orbitrap, so it's a really high resolution equipment as well. So this little ice age is real, and it's preserved in the sediments. And then this goes up to the present. So the temperature range, both in instrument record and reconstructed temperature, is only 1.5 up to 2 degrees. And the little ice age is about 1 degree or even less than 1 degree of change. But it's there, it's recorded in the deep tropics. So if we look at the mean annual air temperature on the terrestrial side, we go from the medieval warm period, and we also have this little ice age. Then it continues to increase to the present. So let's, if we put them all together, the blue envelope is the instrument record, and the mean annual record, which is closest to that area, is within that error record or the range of temperatures measured, at least for the instrumental record. So it seems to work. There are some problems with the proxies, but in this case, it works. For the sea surface temperature, this is the closest we could find that's extended down to 1920s. There are some underestimates, but at least it's within, at least for most of it, we could see the trends. So the other closest area where sea surface temperature was reconstructed is in the Makassar Strait in Indonesia, and this is their temperature reconstruction in purple circles. So similar temperatures, just 1 degree or less in change. So this proxy could work, especially down in Mindanao. The alkinone could work. We could test alkinone proxies as well, but alkinone could probably work way up farther north. So that's one part of my research focus. Another big research for me is looking into analogs for warm ocean and ocean acidification. Dr. Seringan has already introduced this topic yesterday, and they've seen these sites 10 years ago. It was my first time to visit last November. So this is a slide he showed yesterday. In a small area, there's a lot of variation in terms of physical-chemical conditions where you have this submarine groundwater discharge. So this is about 2 meters. The temperature, at least of the water that comes out, is cold or slightly colder than ambient or similar to ambient. You've got the bubbles, you've got a lot of fishes, and there's still corals. Oh. Where you have those vents, if you would look closer, there is this a lot of white fibrous material. These are the bubbles. And those fibrous material are microbial mats, and we are sampling this area. So in a lot of vents, it's always surrounded by mats. One of my students is working on this, and she will work on the DNA and the lipid side. But at least in the literature, this has also been reported in areas where you have high CO2 emanations. It could be thiotrix, or it could be a family of thiotrix, and those bacteria are adapted to high CO2 content or dissolved CO2. In their lipids, they have adaptation to this. They can close and open their lipids just by having slight changes, putting in a double bond or putting in a cycle or cycling one part of their lipid. So this is one part. And you've seen this video yesterday. As you notice, there are no rubbles anymore. This is slightly deeper, 5 meters in depth, with a lot of bubbles, a curtain of bubbles. In this part, where you have less bubbles and the substrate is more stable, you also have seagrasses. So that's me and my student trying to sample the area. So farther away, where you don't have a lot of the bubbles coming out, you have seagrasses. Seagrasses love CO2, and it's shallow enough for sunlight to penetrate. So although it's not diverse, but it's nice to see the seagrasses. I'm trying to count them in one quadrat. I've seen 2 types of seagrasses, and Halophila is one of them. It's very adapted to less saline conditions. So it would be nice to look at their deuterium isotope values in the lipids, because they could be sourced from a different type of water from the submarine groundwater discharge. So we go down just a few tens of meters away. It's about 15 meters in depth, and now you see a different substrate. Temperatures now are very hot, actually scalding hot. You don't notice it at first because when you're diving, it's very, very cold. But you touch it and it's really, really hot. That's the submarine groundwater discharging out. Initial analysis, we've got about 85% carbon dioxide. So you see it's a different temperature, and you see the water curling. In areas where you don't have a high flux of the SGD, you form these mats, which are colored yellow and orange. It's probably sulfidic. So it's a really nice system to analyze. Now we go deeper, and in this case, pressure is greater 3 times. So you would also expect dissolved carbon dioxide is much greater in this system as well. But you'll find, I think there's a video for this. Temperature is also hotter. You see the water curling, but you see fishes in some areas. It's still diverse life forms. So it's a good system to study for ocean acidification and higher and warming temperatures. So one of my students is working on it. He will use DNA and lipid analysis. One of the questions in mind is that this area here in the Philippines is within the Coral Triangle, and one of the questions I'd like to throw out is that what is the role of the submarine groundwater discharge, which from vent to vent could be very different in terms of physical-chemical conditions. What role do they play in the marine biodiversity in this area? And this area is known to be the heart of, or the center of marine biodiversity, and that's one pathway we have not really explored. There are other areas in the Philippines, probably a lot of it with submarine groundwater discharge, but with a different physical-chemical composition. Just outside our marine station in Bolinao, at low tide, you see SGD gushing out. In an area where you don't have a lot of rivers, actually this area does not have a river, you just have surface runoff, that would also mean that submarine groundwater discharge would play a very important role as a conduit. And it's proven in studies by MSI personnel that nutrients in submarine groundwater discharge is higher. It's my hypothesis that it's not just nutrients, it could also be conduit for contaminants, pesticides, pharmaceutical products, or anything that you might find in the terrestrial environment. So at low tide, there's no river, but these are SGD gushing out. And you find them aligned, so it's really a big, significant factor we need to consider. So one of the questions I'd like to look at is, aside from looking at the chemical composition of SGD, is that the question, could SGD play a role in triggering algal blooms? So I'd like to go back to those places that Dr. Rodora Asanza has looked into and check if they're SGD and what are the physical chemical compositions of this SGD. So the fourth major topic I'm working on is blue carbon systems. How they of course, they sequester carbon, they store it, and then they export it. The blue carbon economy is very active right now, but there's still a lot of items we have not pegged in the blue carbon economy. So these systems include the coral reef, seagrass meadows, mangrove forests, and salt marshes. These coastal areas have very, very high primary productivity. In some parts of the open ocean, in contrast to this, you have low primary productivity. So these coastal areas, in some cases, might be able to subsidize heterotrophic activity in the open ocean, and that's what I'd like to look at. So it would start in carbon sequestration, and what's the carbon stored in the system, and eventually what's exported. What's exported, what form is it in? Is it as inert like black carbon, or is it very labile and very active in the biogeochemistry of the ocean? So are they able to subsidize heterotrophic activity in the open ocean? So these are my students working on blue carbon areas. This guy is working on mangrove area. Another student working on seagrass areas. And thank you very much.
We have plenty of time for questions. Any questions? Yeah.
I'm not an expert in biology, but I heard that corals are very sensitive to water temperature. So, like, does the hot water from the ground discharge do, or does not affect the coral reef ecosystem?
Well, we know it's always reported that corals are very sensitive, but as Dr. Siringan also reported yesterday, some types of corals, like the Heliopora, they seem to be more adapted to more extreme conditions. So we don't totally understand yet how other corals are able to cope. But in these cases, there are still corals around the area. So it's a very interesting place that we could survey and we can try to understand how the corals are able to adapt. Are they thriving, or are they just surviving, and what types of corals are there? So it's a proposal stage right now, and that's part of the proposal that Dr. Siringan is leading.
But no bleachings or no destroying the coral systems recently found yet?
We still have to survey that part. In this place where, well, it seems consistent that you have CO2 emanating for so long in that area. It's a slightly different coral cover. So this might have already selected the type of corals that are already thriving there. So there's no, at least in the small area that I've seen last November, there's no bleaching, but they must have been the corals that's already survived these conditions.
Okay, thank you.
More questions?
This is not a question, but probably a suggestion. The other question was regarding the impact of the groundwater discharge on the coral reef ecosystem. I think you can do some experiments in the lab to test the materials that you get from the groundwater discharge and get some of the materials that you encounter in the area. And also you may try doing that for the endosymbionts of the corals, the zooxanthellae, and they can be cultured. Thank you.
Thank you very much.
Professor Lee, yeah.
You showed us some the surface temperature proxies, for example, the GDGT and the Arcanon. Maybe Arcanon is not working there because the surface temperature is a little bit higher. So my question is two. One is you showed the variation in terms of depth. Do you already control the age for these cores? That's the first question. And second question is, I think it will be interesting if you can compare the SST variation to the precipitation history in this area. Do you have a plan, or did you already do that?
Thank you very much for your question. I wasn't sure if I have enough time to discuss it. So this core has been age dated using carbon-14 pollen material and lead-210. So this is the lead-210 part. And then I did not plot it here, but there are about 8 carbon-14 age dates in the core. So we're quite confident with our age model. I'm working on the sea surface temperature reconstruction, but actually this is a project that Dr. Seringen and I are collaborating on. And he has students working on the inorganic geochemistry of the core. His students has looked at titanium, and it's eventually in our plan to look at precipitation proxies. Mainly, we use titanium and ratios of titanium, aluminum, silicon titanium, or zirconium titanium. So eventually, we would like to compare that from the marine side and from the terrestrial side. So both cores were dated, and then inorganic geochemistry or XRF was done on the core. Yeah, so it would be nice to get this out and published at some point.
So the core is the length of core is about 1 meter.
1.4 meters.
It corresponding to the how many years?
Up to 1260 AD. So about 900
years.
Yeah, 900 years. And this might be very interesting for you because it's very close to the area where you want to work on. So this is on the this is where the Mindanao current goes through, right here.
Yeah.
So from our discussions last night, I think you're interested to core in this area, and if there's a typhoon, we go here, or this is Davao on the other side. So yeah, it would be nice to see a very long core at some point, and then have a very long data
Okay.
on the area.
Thank you.
Okay.